a different kind of standard, should I say?
Yes, indeed. Well, good afternoon, and thank you for the gracious invitation to speak today. I'm a consultant on manufacturing and regulatory aspects of cell and gene therapy for Slingshot, and I've been asked to, I'm pinch hitting today, but I'm actually quite pleased to do so, since Slingshot's technology is quite exciting, and I've thought it was enormously promising since the very first time I heard of it. Two of my slides are from Excelix, so I've brought along a cheat sheet to make sure that that I get those slides right. So wanted to talk a little bit, and thank you, Ruud, by the way, for pretty much the ideal introduction to this talk. I couldn't agree more about the need for better standardization and better control and the limitations of the existing biological controls that we have. Slingshot's technology is designed to address those problems and does so, as I propose to show you in the next few minutes, quite beautifully. I think we can all agree that we need controls for our manufacturing process, for in-process testing, for product release, analytics that are better than our existing biological controls. Our existing controls are considerably limited. We need them for analytical validation, for in-process and release QC testing, for comparability, perhaps the single biggest challenge in cell therapy. And of course, there are just plain regulatory requirement. And we have two sources of biological controls, both of which have substantial flaws: cell lines and primary cells, donor-derived material. Both of them have substantial flaws. They're complex, and sometimes they are just plain unavailable. One, there are variability, limitations in data quality, impair assay validation, they can delay product release, delay patient care. There's also, they also are associated with a substantial resource burden, whether it's sourcing donors, developing, maintaining cell lines, doing bridging studies from one biological control to another. They're also quite costly. Tech transfer can be delayed due to challenges with distributing your biological reference standard among different sites. As Ruud pointed out, different sites also may be using different analytical technology, different flow cytometers and such. And of course, bad data leads to repeated studies, which has time and dollar costs of its own. And sometimes the right biological material is just plain not available, whether that's material that has the rare biomarker that you're keenly interested in studying, rare donor material and such. And you can get a pretty good numerical idea of this, even though these data come from 3 different papers. Here we see %CV data for different mononuclear cell subsets, for %NKs, CD56, %CD19, CD3, CD4. They're from 3 different studies. But you'll notice that in all cases, the %CV, these are data for cells analyzed prior to selection on the Clinimax. So the data happened to be published, and so we took advantage of its availability in the published literature. And you can see really pretty appalling CVs. 42% in the case of NKs, 39%, 13% for CD3s. Well, at least that's a little bit less, but still, as a clinical pathologist, I have to say a CV of 13% still, you know, makes my stomach churn. 42% for CD4. So really just plain not good enough. So, okay, what if you turn to cell lines? Well, cell lines come with problems of their own as controls. For one thing, you have to maintain them. You need to build a master cell bank, and it either needs to be a really big master cell bank, or you need a tiered system of a master cell bank and working cell bank. That's not commonly done in the analytical setting. And so oftentimes, to avoid depleting the master cell bank, what one ends up doing is culturing the cells for perhaps too long, leading to antigenic drift. But frankly, even if one doesn't culture the cells for too terribly long, antigenic drift can show up surprisingly quickly. Here you see some internal data from Slingshot for CD19 antigen density on Raji cells cultured for just 6 passages. And you can see really quite substantial changes in antigen density. So ideally, we would want a control that's engineered to have the same amount of antigen expressed, the same cell surface markers, the same antigen density in every single lot, every single day, that we could distribute to different clinical sites, regardless of what analytical instrument they're using, with the assurance that this stable, consistent reference standard would be common throughout all of our different sites, or even at one site would be consistent from June to January and so forth. And that's really where Slingshot steps in. This is a new kind of control intended to take the place of traditional biological controls with considerable superiority. Slingshot calls these cell mimics. I think of them as almost synthetic cells. These are precision polymers that are manufactured using semiconductor technologies and inorganic materials that are intended to address the variability and stability limitations of biological controls. And Slingshot adds in, decorates them with biological features. They can add the necessary biological characteristics, cell surface markers, and other biological features that give their cell mimics very high lot-to-lot reproducibility, eliminate issues with cell line maintenance, and eliminate biological material sourcing, give you predetermined levels of antigen, allow one to place rare biomarkers as desired, with full customizability and greatly facilitating analytical validation, tech transfer, comparability, and so forth. When I first heard about this technology, I was really quite impressed, and this was one of the rare times when I actually approached a company and said, please let me consult for you, because I would like to help make sure that that your technology is successful, because God knows we need it. I've been working in cell therapy for 30 years, and I've seen far, far too many promising cell and gene therapies fail simply because of problems with inadequate or unavailable reference standards, problems with tech transfer, problems with comparability. So it's really quite a beautiful thing. And as I learned more about it, I became more impressed. It's not just that they can place cell surface markers, although that's cool enough by itself. They can also place intracellular proteins inside these precision polymers in a way that makes them really quite superior to the kinds of plastic particles that we're used to putting up with as our next best, as historically our next best alternative. This is something that is much, much more cell-like, yet it is consistent and highly scalable, quantitative and precise, and extremely stable. So you can pull something in July, and be sure that it's consistent with what you pulled in January. It's very flexible and customizable, and my favorite word, well controlled, and ISO 9001 certified. So quite beautiful polymer cell mimics. Here we see data for an example of Slingshot's TruCyte phenotyping controls. Here, the dot plots in orange are TruCyte's lymphocyte subset cell mimic, and this is designed to mimic the normal ranges for T cells, B cells, and NK lymphocyte subsets. Cryopreserved PBMCs are shown in blue. The TruCyte lymphocyte subset cell mimics are shown in orange. And you can see that the TruCytes show tighter and greater antigen expression, superior lot-to-lot consistency and reliability compared to the biological controls that we've had to, well, put up with over the years. I really have to echo everything that Ruud brought up in terms of the difficulties of biological controls, as he described them so nicely in his previous talk. This really is quite a lovely solution to that problem. And you can get a handle on that just taking a look at lot-to-lot reproducibility. When you look at data for CVs across 2 lots of each control type. Here you see data for 3 types of commercially available controls: lyophilized PBMC lots, cryopreserved PBMC lots, lyophilized lymphocyte lots. Sorry, my tremor's fighting me a little bit today, so apologies if you get a little bit seasick when I turn the laser on. These 3 are shown in these columns. Slingshot's TruCytes, TruCyte lymphocyte subset CVs are shown in this column. So whereas the monocytes for example, cryopreserved PBMCs, the CD45 percentage has a CV of 34.5. For TruCytes, it's 4.4. For CD3 positive cells, okay, well, that's close, 1.2 and 1.3. For CD19 positive cells, for B cells, a CV of 19.8 for cryo PBMCs, only 5.7 for TruCytes. Across a number of common and important biomarkers, really quite tight CVs compared to the kinds of commercially available biological control material that we have to deal with, that we've had to deal with until now. And now a couple of brief case studies. How am I doing on time? More or less okay. This is a collaboration that Slingshot undertook with the Fred Hutch Cancer Center. The Fred Hutch is doing EGFR positive CAR T cell therapies and needed an EGFR positive T cell mimic. On the left you see their data for transduced human T cells. On the right, you see data for untransduced human T cells that were spiked with Slingshot's EGFR positive mimic T cells. And you can see that, again, we see tighter gating and a very nice, clear separation of the population. The TruCytes, the EGFR positive Slingshot cell mimics were engineered by Slingshots specifically to address the variability in donor-derived CAR T cell assay controls to give us greater stability and consistency. The standardization allows you, or allows the Fred Hutch in this case, to have better quality control, better reproducibility, supports comparability, and according to Fred Hutch, they achieved about a 60 to 75% savings by minimizing or eliminating the need for in-house maintenance of biological control material. Quite a nice result for them. Now, so far, we've talked largely about cell surface markers, but in fact, it's possible to use Slingshot cell mimics to look at intracellular markers as well. In this case study, we see results for use of TruCyte cell mimics looking at the nuclear envelope associated cell activation marker Ki67. This was done in collaboration with SERBA. SERBA Research provided dot plot and histogram data of their desired Ki67 expression. We see that here. The cell mimics shown below. Oops. There we go. The cell mimics shown below. Again, apologies, the tremor is fighting me today. There's only so much that propranolol will do to cut it. And so here we see dot plot and histogram data of SERBA Research's validation of Slingshot's Ki67 mimic. Again, quite clean data, and for an intracellular marker. So a very pretty thing indeed. Now we come to Slingshot's collaboration with Excelix, and here I need to refer to my notes more. Slingshot and Excelix have entered into a collaboration, and in many ways the two technologies are made for one another quite nicely. Slingshot's cell mimics can be used with any flow cytometry platform, including that of Excelix, but the two are particularly nicely matched up in that the Excelix platform is a highly automated benchtop flow cytometry, fanless, very easy to use, intended to be to require little or no, no calibration. Excelix assays are intended to be off-the-shelf. Custom assays are available. They're cartridge-based. They use a dried-down format. And so this is kind of exactly the circumstance where you would not want to use a traditional kind of biological control. Having to use cryopreserved PBMCs would in many ways ruin the whole point of Excelix, or would take away a lot of the advantages of using Excelix's technology. Applying the Slingshot cell mimics in this case really makes a great deal of sense. Slingshot's TruCyte controls were applied, positive and negative controls. These are stable. They gave extremely reproducible results in Excelix's evaluation. Let's see. A couple of other details about the Excelix technology. The single cartridge assay has a cartridge and reagent tube with a dry antibody cocktail. 40 microliters of sample is added to the reagent tube, mixed for 2 minutes. The Excelix instrument recognizes a QR code on the cartridge, runs the appropriate protocol for sample prep, data acquisition, and analysis. Ruud, you were talking about wanting more automation. I give you this. So this is the kind of thing that we need to see more of. Far too much of the technology that we use even today in cell therapy analytics is not that different from what we were using 25 years ago when I was training in cell therapy. The assays are automated, are algorithm-based, gating is consistent, so you get away from manual analysis and all of its attendant variability. So using the Slingshot TBNK, T-cell, B-cell, and K-cell TruCyte controls are kind of ideal. And on the next slide, here we see FlowJo scatter plots of the TruCytes tested with the Excelix TBNK16 and T-cell assays. And in general, you'll note the tight gating and clear separation of populations that we saw in the previous flow cytometry assays, flow cytometry slides. This was also seen in Excelix's stem cell assay data. That's not shown here, but for data on that and more data on the Excelix TBNK and T-cell assay results, you should come and see two very interesting posters from Excelix and Slingshot at the poster session this afternoon. That'll also have more information about application of Slingshot cell mimics on the Excelix platform and more information about Excelix cell mimics in general. So certainly the results would seem to indicate that the TruCytes biomarker controls work well with the Excelix assays, Excelix platform and Excelix assays, and I would argue this by extension means that they have broad application in cell-based analytical platforms in general. But the Excelix platform seems particularly well suited. Taken together, the Slingshot cell mimics and the Excelix platform are a beautiful kind of all-in-one solution to a tremendous amount of our analytical dilemmas in cell therapy. Now, so far we've talked really about flow cytometry. One of the things that that I got very excited about was when Slingshot showed me results obtained in functional potency assays, however. And here you see data for use of Slingshot cell mimics in a BCMA CAR T-cell potency assay. The Slingshot cell mimics were used to stimulate BCMA CAR T-cells in an interferon gamma release assay. It's nice. It's extremely encouraging to see that that these cell mimics can be used in bioassays, such as in interferon gamma release. I would point out in particular that when TruCytes are employed, there's measurable interferon gamma release in as little as 6 hours. So you have opportunity for more rapid turnaround. Okay, heck with it. The trimmer doesn't allow me to point that tightly, but you can see the blue vertical bar at the 6-hour time point. The orange and amber vertical bars. The blue vertical bar for the MM1S cell line. Notice how large the CV is there. 32, representing 32.6%. By comparison, the TruCyte cell mimics at that time point have a CV of 7.99%, quite tight indeed. They continue to be very tight, 11.6% at 24 hours, 3.95% out at 48 hours. Overall, CV never gets above 12%. Whereas with biological cells across different passages, we see percent CVs of up to 33%. Wrapping up, Slingshot Biosciences in general in the cell therapy process, what I hope I've indicated to you is that there's opportunity and good reason to use Slingshot cell mimics at multiple points in cell therapy manufacturing and testing, whether it's characterization of apheresis products, characterization of patient material and rare biomarkers, testing of isolated cells, testing of expanded cells, release testing, ongoing patient monitoring, and so forth. There are multiple places where Slingshot's technology, where Slingshot cell mimics, I think, have a place to be very productive and useful in the cell therapy process. And last or second to last, There's also very good regulatory support for this approach. Multiple FDA guidances allow for the use of alternative reference material in development, validation, and deployment of analytical methods. In particular, Q2R2, that's a May 2024 update on assay validation, and I'll quote from that guidance: reference material or other suitably characterized materials with documented identity, purity, or any other characteristics as necessary should be used in the validation study. Again, or other suitably characterized materials. So certainly there is regulatory support for this approach across multiple guidances. Last of all, and I swear to God it's last, Alex. Last of all, because TruCytes reduce the need for in-house qualification of donor-derived primary cells or cell line-derived control materials, one can realize considerable savings in both time and money, in addition to while at the same time achieving greater consistency and greater control, reducing variability, and getting to the greater precision and accuracy that we need. With that, I thank you very much for your attention, and I think we've got a few minutes left for questions. Couple minutes left.
Have we? Yes, one question.
Hi, I'm Jyoti. I am from Excelix. Nice talk, Scott. So regarding the Ki-67 mimics, which have a CAR-like profile, how do the CVs for that kind of a product look? Because all the other CVs you presented were for...
I'm sorry, I'm having just a little trouble hearing you. Can you, one more time?
So the product, Ki-67...
Can you state your name and company?
My name is Jyoti.
Yeah. I can actually hear you better now.
So the product Ki-67 mimic, which showed the CAR-like marker, right? CAR-like marker?
The Ki-67 was the nuclear...
So how the CVs for a batch-to-batch reproducibility for a product like that look?
Let's see. I don't think I have data. I don't think I have data on...
Follow up with you later.
Okay. I don't think I have batch-to-batch CV data for that.
I'll follow up later then. Thank you. Something like that, yes.
I think I only... again, apologies, I'm pinch hitting here. And I think what you need, the person you need is actually a Slingshot manufacturing person. But I will ask Slingshot to follow up with you on that. Actually, someone from Slingshot must... Ah, yes. Can someone from Slingshot tell us? If you happen to have a number off the top of your head.
This particular control was made for Cerba. It was a single lot. So there isn't really data on the CDs for this particular control, as it was just a one-time use of that.
Well, then in that case, you see my answer is, it was a single lot.
Awesome.
Thank you very much indeed.
Thank you, Scott and Ruth, for those two very nice talk about the importance of having controls and well-characterized controls, not any controls. So I'm not going to talk too long because I stand before in between your lunchtime. So there, we can reconvene at shortly before 2 o'clock here for additional talks by Ben Espen